I'd like to talk first historically about the last 30 years of artificial transcription factor research and then what we're doing currently. What I won't be talking about today is any of our functional genomics applications with CRISPR-I or CRISPR-A because Jonathan and Max, my co-conspirator, will talk about that tomorrow. So that's why I have room today to talk about a little bit more old stuff, which I actually am very enthusiastic about, and I think we can. I think it's worth reading old literature and learning from mistakes and insights that have been now forgotten for some things. Okay, so a quick outline of the talk. I'll give an introduction to transcription, just to make sure we're all on the same page. I'd like to talk about work from the 80s where they defined what an artificial transcription factor could be, and that this went a long way to defining how transcription factors actually work in their endogenous setting. Then I'll talk about the next 15 or so years where people used artificial transcription factors with programmable protein-mediated DNA binding, and then talk about our current efforts repurposing CRISPR as an RNA-guided artificial transcription factor, and then some additional applications at the end that I think are equally cool, but are have a ways to go. So Jennifer showed this essentially same slide. The classic central dogma of biology is that our genomes encode genes. This information is transcribed into RNA. RNA is made into proteins, and then the proteins carry out functions within cell that both replicate DNA and keep cells alive. We all have lots of cells in our body that have chromosomes. Our chromosomes contain both coding and non-coding information. The coding information is turned into RNA by RNA polymerase in concert with additional general transcription factor machinery and also sequence-specific transcription factors. And so the general transcription factor machinery was worked out first for the most part. RNA polymerase is a conserved complex from bacteria through to us. We have 3 RNA polymerases where bacteria have 1, but you can see from this crystal structure that the general features of this machine are conserved across all life. Archaeal and eukaryotic RNA polymerases have additional subunits. These are important for a variety of specialized applications, but also generally human RNA polymerases are required to transcribe genes which can be many up to 100 kb long, which isn't the case for bacteria, and so it's possible that there's increased processivity that's encoded into these additional subunits as well. So I think the central question in transcription that we've been working on for essentially as long as modern biology has been around is, and we continue to work on, is how does a cell know when and how much of each mRNA to make? This is a really fundamental problem to all life, but especially is an important problem in multicellular organisms where we have many different cell types across our body that do different things. And how do you, within a specialized cell, execute one program and in another cell execute another program, and how is this orchestrated during development? And what happens when this goes wrong in disease or during aging? And so really this boils down to sequence-specific transcription factors bind sequences encoded in our genome to regulate gene expression, but we still don't understand really how this process works for many cases. And there are a few well-worked out, canonical examples that we know a lot about, like hemoglobin transcription, but for the most part, for your average gene, we know very little actually about how its expression is controlled. And so just to give you a sense of this problem, if we look at the simplest bacteria, which is this mycoplasma, it has one cell type. It has a small genome, less than 1 million base pairs. It has 517 genes, and it has no transcription factors. So it has RNA polymerase and a couple RNA processing proteins, but there are no transcription factors that look like a standard transcription factor in this genome. Everything's just transcribed constitutively all the time. That's sort of boring or amazing, depending on how you look at it. In yeast, things are a little bit more complicated, but still manageable. And so for a long time, we've used yeast as a model for eukaryotic transcription. Yeast have roughly, depending on who you believe, about 169 transcription factors. But there's still just one cell type, and they undergo specialized processes like sporulation and meiosis, which are complicated, but it's still fundamentally one cell type. Whereas estimates vary, but most of us in the room probably have about 200 cell types, hopefully not more. But you can see that our the number of annotated transcription factors varies depending on who you believe, but is expanded at least tenfold above what's present in yeast. And so most of these have no known function. So like the biggest class is the crab-associated zinc finger proteins that we'll talk a fair amount about today. And there are about 400 of these encoded in the genome. It's sort of thought that they're involved in silencing retrotransposons. They may be involved in speciation. It's one of the biggest class of expanded proteins in primates. But really, there's a lot known about one or two of them, and the other 400, there's like very, very little known, even though it's known that they bind across the genome. So I think the big pictures the big considerations that are unsettled in transcription is that there's this complicated interplay between general transcription factors such as the initiator complex and the mediator complex, and sequence-specific transcription factors which bind at cis-regulatory regions and promoters and enhancers, and how those interact to mediate the output of an individual gene is still an active area of research. There's lots of feedback loops involved in regulating both transcription factors, which are master regulators of specific developmental processes, and these things have negative feedback loops and positive feedback loops, and transcription factors go on and off, and the dynamic regulation of both transcription and the transcription factor network is important for pretty much every process in your body. And then I think still a very active area of research, and one that's been continues to get more interesting with a lot of our capability to sequence many millions of reads, is how the genome is actually organized to control transcription. So the genome is not a linear sequence of nucleotides. It's actually a 3D structure within the nucleus. There are areas of heterochromatin, which are shown in this lighter area of this picture, and there are darker, and there are areas of euchromatin, which are shown in lighter areas, and these are known to broadly correlate with expression or lack of expression of genes. But how an individual chromosome is folded within the nucleus is completely unknown. And what localization does to gene expression is still, I think, a very open question. And then as a sort of part B to that point is that local chromatin state has major effects on gene expression. And we know sort of a lot about this, but there's a lot we don't know. So how do we get at this? So there's sort of two ways. You can take snapshots of what something is doing. And so these are things like ChIP-seq studies where we can measure the occupancy of a transcription factor across the genome and correlate it with gene expression and say, where we see this transcription factor bound, we see high levels of this histone mark, and we see active transcription. And so this transcription factor must be correlated with active transcription for this class of genes and repression for this other class of genes. This speaks to one of the points I raised on the previous slide. There are two layers of genetic control or two broad layers of control over transcription. So, and they're sort of artificial. People classically think about genes having promoters, which are defined as the sequence, usually 5' of the transcription start site of a gene, where you have binding sequences for general transcription factor machinery, such as the TATA box, but also upstream elements that are involved in activation or repression of gene expression, which are recognized by sequence-specific transcription factors. And then there are enhancers or silencers, which are usually cis-acting sequences, but can be very, very far from a gene. And it's thought broadly that both are important for gene expression, and that there's a three-dimensional organization of the genome that often brings enhancers, which are up to 1 million base pairs away, actually quite close to the transcription start site of a gene. And this chromosome looping is thought to be dynamic, and it's thought that this is critical for the way gene expression is regulated. And so that's sort of diagrammed in this cartoon. But really, a lot of the players involved and how they're regulated is really not clear. But it's even more complicated than this, in fact. So DNA doesn't exist alone. It's wrapped into nucleosomes. Histones have tails, and there are lots of marks deposited on these tails. And these, the orchestrated readout of this governs gene expression. And so really, in the end, what you have is this huge number of proteins that are involved in a dynamic process that controls the output of the genome. And we've studied, we know a lot about a handful of examples of histone acetylases and demethylases, but for the most part, there's a lot left to learn about chromatin biology and about transcription in general. So that's all a plug for this point, which is what I was trying to say earlier is there's two ways you can study this. You can study it by correlating where proteins are across the genome and the output of the genome, or you can take more of a engineering approach and say, okay, if we artificially engineer in sequences and proteins that are not found in nature, what does this do to transcription? Or can we take a sort of a reductionist approach or an engineering approach to decouple how a genome is regulated? And so these are sort of impossible to decouple. You really need both approaches and both approaches have been very fruitful over the last 30 years. And I'll talk initially about artificial transcription factors with fixed DNA binding specificity. And this is work of many people, and for the sake of time today, I'm really only going to talk about the work from one lab, which is Mark Potashny's lab. And he was one of the early pioneers in this field, but there are a lot of other people, including people at Berkeley, who are also pioneers. But in the context of chimeric artificial transcription factors, I think there's a fairly cohesive body of work from his lab, and so that's what I'll talk about today. So what is a sequence-specific transcription factor? It's a modular protein, is one definition. The other critical feature is that all of these things do what I've said they do. They all bind to DNA. So all transcription factors have a DNA binding domain. Beyond that, there's a lot of diversity in what they do. They can be repressors, they can be activators, they can act as scaffolds to recruit additional enzymatic activities, such as histone acetyltransferase activity or demethylase activity for either DNA or histones. They can directly recruit basal transcription factor machinery. So we'll see throughout the rest of the talk that one of the most commonly used chimeric transcription factors just directly recruits the mediator complex. And then one thing that I'll show you actual data for is that what was realized very, very early on is that you can split the DNA binding activity from the effector domain activity and create chimeric proteins that have one DNA binding specificity and an arbitrary activity. And so if we go back all the way to 1982, when people still worked on these things in E. coli, there were sort of two models for how transcription was controlled. So model one says that transcription factors bind DNA in a way that physically stabilizes one orientation of a DNA structure such that basal transcription factor machinery can bind and activate transcription of a gene. And so this is one model. The other model, though, is perhaps more complicated and interesting to me, is that people thought perhaps transcription factors actually bind to DNA and act as a scaffold to recruit additional proteins. And so there was some evidence for model 2 that was from the early 80s, where people were studying the lambda phage, and they knew that this lambda phage protein called repressor could both activate and repress genes, and that, so that's diagrammed here, where repressor is bound to two different elements and it dimerizes. It represses the expression of a lytic gene required for the phage lytic cycle, and it activates expression of its own production. So it activates expression of repressor. And what a paper in 1982 claimed was that you could generate mutants of this protein which still repressed expression of this gene, but no longer activated expression of this gene, suggesting that this gene was still binding and repressing the lytic genes, but there was some critical interaction that could be lost that broke the ability of this repressor protein to induce its own expression. And so this, in a way, suggests that you can make a mutant that is no longer able to recruit things that are required for expression, but really doesn't settle the issue. And so this was published in '82 by Mark Patashny's group. But I think there was there's a lack of clarity in the field, and people thought, well, this is phage biology, and that's pretty simple. How does this actually work in eukaryotes? And so what the lab at the time did was turn to what was at the time a fairly well worked out system, which was the yeast Gal4, Gal1 system. So in yeast, there's carbon sensing, and the Gal genes are regulated depending on whether the yeast is seeing glucose or galactose. So in the presence of glucose, expression of Gal1 is turned off. In the presence of galactose, Gal1 expression is turned on. And this is mediated by a transcription factor called Gal4, which binds to a Gal4 UAS sequence just upstream of the Gal1 gene. So when it's bound and it's not bound to its inhibitor Gal80, Gal1 goes on. And so this is a nice dynamic setting in which we know some of the proteins involved, and we can start to decouple whether a transcription factor is recruiting other proteins or whether it's just stabilizing a DNA structure. And so the experiments that they did are summarized here. This is, as far as I can tell, one of or maybe the earliest in eukaryotes domain mapping and domain swapping set of experiments that's been done, where they, in a series of experiments from 1985 to 1987, made a bunch of deletion mutants of the Gal4 protein and asked whether, with reporters, they could still get DNA binding and whether they could still activate transcription. And so what they did was they cloned a Gal4 UAS sequence upstream of a reporter gene and made a bunch of truncation mutants and asked whether they still got DNA binding and whether they could still activate expression. And with this, they were able to show that these two activities of DNA binding and transcription activation are separable, and that you can separate them. That there are some that essentially they showed that the N-terminal 74 amino acids are required for DNA binding. So this is the DNA binding domain, where there's two domains within the protein defined by these internal deletions down here that are required for gene activation. So this is really the first example where somebody systematically went through and defined that transcription factors are modular and that you can separate these activities. I wanted to show the primary data because it's actually hilarious. This cartoon is just a cartoon, but this is the actual data that went into the cartoon, which is also just a cartoon. And so it's funny if you read these old papers frequently, there's huge amounts of data not shown or things summarized without any primary data, which I think wouldn't fly so much now, but it has stood the test of time, so good for them. Equally interesting, I think, was this other paper from 1985 in which, to my knowledge, this group created one of the very first, at least in eukaryotes, artificial transcription factors. And so they were, again, still trying to get at this idea of, can you decouple DNA binding from effect on transcription? And what they did was they fused the N-terminus of Gal4 to a bacterial transcription factor called LEX-A, to create a fusion protein where the Gal4, most of Gal4 is now attached to an E. coli DNA binding protein. And then they clone a bunch of reporter constructs shown here where they have different upstream binding sequences and different promoters, and looked at whether you got expression of these laxity reporters in the presence or absence of glucose or galactose. And what they could show was that if you cloned a binding site for this LEX-A DNA binding domain and added either glucose or galactose, you got activity, suggesting that this fusion protein can constitutively bind to the LEX-A UAS element and induce transcription that was decoupled from the nutrient sensing in yeast. So in this setting, glucose and galactose sensing occurs at both the level of the GAL4-GAL80 interaction, but also at the level of GAL4 DNA binding. And so they could show that if you had yeast in a galactose-containing media, this was bound and was activating LAC-Z expression. But also, if you had just glucose in the media, you have constitutive binding of this artificial transcription factor and induction of the LAC-Z reporter. And so I think in its own crude way, this is really the inspiration for all the work that's been done over the next 30 years in eukaryotes, where we've made increasingly complicated artificial transcription factors. But really what they wanted to do was figure this out, but also the lab went on to create a variety of tools where you can artificially turn gene expression on or off. And that's what we're still doing. And it's gotten more complicated and easier to use, but this is really the heart of it. So over the next couple of years, the same lab and other labs asked the question of whether this was also true in human cells. So they had shown it in yeast, which is a eukaryote, but it's always possible that things are more complicated the higher in evolution you go. And so pretty much at the same time, two different labs published that this same modular chimeric artificial transcription factor approach could work in human cells. So what both labs did was express the yeast GAL4 DNA binding sequence fused to an activation domain in human cells, and then look at whether you could activate a reporter. So one lab fused GAL4 to the estrogen receptor or the glucocorticoid receptor, as shown in these cartoons, and then added either estrogen or dexamethasone. And you can see that in the absence of estrogen or dexamethasone, you get very little transcription of your reporter. And then when you add the inducer or the activator of the estrogen receptor or glucocorticoid receptor, you induced expression of a reporter. At the same time, another lab cloned an even stranger artificial transcription factor in which they fused also the GAL4 sequence to a herpes virus protein, this VP16 protein, and showed that this was an unusually potent transcriptional activator when targeted to a repressor that had the GAL4 DNA binding UAS element either close to the transcription start site of the gene. It was a potent activator if you targeted it close to the transcription start site, but this was the first suggestion that in human cells, that positional recruitment of a transcription factor was critical for its activity. So when you recruit this artificial transcription factor here, right next to the transcription start site, you see a huge amount of activity of your reporter or activation of your reporter. But if you recruit it either 1 kb upstream or roughly 1 kb downstream, you see a lot less activation of your reporter. So the lesson from this paper was essentially, you can create an artificial transcription factor in a human cell. It will bind and activate transcription, but that the positional rules apply, and this was known from yeast actually, that the activity of a gene is dictated in part by where a transcription factor is recruited relative to a transcription start site. This work with this GAL4 VP16 fusion is actually maybe still being used by people in this audience today. So this was the sort of the first experiment that eventually led to the creation of what are known as TET-on and TET-off systems. So a lot of you use inducible promoters, which are sold commercially as from CloneTech mostly, as TET on and TET off, where you can inducibly control expression of your gene of interest. And really what those systems are an artificial promoter that contains a TET, so 7 copies of a TET repressor binding sequence upstream of a minimal CMV promoter. You then clone your gene of interest and express it in your cell. And you co-express a TET-R VP16 fusion protein that when you add doxycycline or remove doxycycline will bind to the promoter and activate transcription. And so this was, it has been a very useful tool in a lot of different fields, because you can get really strong expression and it's in the right context not very leaky. So that's shown here, where you can get 5 logs of expression depending on whether you have or don't have doxycycline in the media. But really this is essentially no different than the experiment that was done in yeast. It's a bacterial transcription factor fused to a viral protein expressed in a human cell with a recombinant or an artificial promoter that you add a chemical inducer to. It's like completely synthetic, repurposed from other organisms, but transcription is conserved enough that this works quite well. So just a brief foray into what is actually going on. So you can use these systems without understanding how they work, but it has been worked out how this actually works. So there are multiple types of activation domains. There are basic activation domains and acidic activation domains and neutral activation domains. But the ones that most people use and the ones that are the most potent are called acidic activation domains. A really interesting experiment done in the late 80s was the same Patashny lab took randomly sheared E. coli DNA and fused it to the Gal4 DNA binding domain and expressed these random non-natural fusions targeted to a reporter, and showed that about 1% of these randomly generated peptides could activate transcription in yeast. And so this was really surprising, I think, at the time. And I think probably nobody believed this poor grad student that did this experiment. But when he went back and sequenced the clones that were activating transcription, he saw that invariably they contained a bunch of highly acidic amino acids. So he had created a Gal4 binding fused to an acidic domain. And this was sufficient to activate transcription in a eukaryote. And through a lot of work in the last 20 years, people have shown that most of the endogenous transcription factors, like NF-kappa B is a good example, have highly acidic domains that directly interact with the mediator complex, and when bound to DNA spaced appropriately from a transcription factor start site, these transcription factors will recruit the mediator complex that then recruits RNA polymerase, causing the promoter to produce RNA for whatever gene you're talking about. And people have now solved the crystal structures of these, and what it is clear is that you can have acidic sequences that insert into a hydrophobic furrow that is at the interface of the mediator subunits 15, 17, and 25. And this interaction seems to be conserved in yeast and in human cells and in natural transcription factors and in viral proteins and in completely artificial transcription factors as well. So it's a very general principle. All that's important is that you have an acidic sequence oriented in the right direction relative to DNA, and that you're spaced appropriately from the transcription start site. So activation is one thing, but just as many transcription factors function to keep genes off as to keep genes on. And so people in the early 90s were interested in figuring out how this worked. And there have been a lot of repression domains identified in natural transcription factors. A lot of this work was done in Drosophila. And what I'll summarize just for the purpose of time is that one of the most potent repression domains that has emerged and what's present in pretty much all of the synthetic transcription factors that people use is a domain called the crab domain. So I referred to this earlier. There are a class of transcription factors encoded in our genomes that are called zinc finger crab transcription factors. There's about 400 of them. Most of them have a CRAB domain. It's a fairly short domain. This cartoon is one of the original, is from a paper where they defined the domain. Essentially, on zinc finger CRAB proteins, there's an N-terminal CRAB domain that has a CRAB A box and a CRAB B box, and different zinc finger proteins have slightly different CRAB A and CRAB B boxes, but for the most part, they all work the same. And this paper showed that you could fuse Gal4's DNA binding domain again to a CRAB domain, and when targeted to a reporter, you could really efficiently repress transcription. So these dark circles pretty much completely go away, suggesting that in human cells you can create an artificial transcription factor that really efficiently can turn transcription off. And as with the Gal4 activation domain, people did a series of domain analysis where they deleted different parts of these domains and defined which domains were required, and then in subsequent work defined how they worked and what proteins they recruit and whether they're context specific and whether there's positional effects relative to the transcription start site. And so that's all been worked out now. Things over time have gotten more and more complicated. So I'd like to just end this portion of the talk by talking about two recent papers which use artificial transcription factors with fixed DNA binding domains to study either heterochromatin memory or formation. And so this paper from 2012 used two inducible artificial transcription factors where they've into the mouse genome knocked either 12 or 6 copies of this zinc finger binding site or a Gal4 binding site. They can then express in ES cells 4 proteins. So they're expressing a zinc finger FKBP fusion and a FRB HP1 alpha fusion, as well as this plant system, which is can't read that from here, ABIPYL1 fusion. Both of these systems are chemically inducible dimerization systems where you can add a small molecule and these domains will dimerize and recruit the domains to the sequence of DNA where you've integrated your fixed DNA binding sequence. And so they showed that you can use this system to look at the timing of recruitment of different domains to study how transcription and chromatin modifications interact with each other and how the deposition of chromatin marks that are associated with heterochromatin formation are maintained and relate to the level of transcription of a specific gene. So this is pretty complicated from where it started, right? Like we went from one artificial transcription factor in yeast to 4 proteins expressed that are independently inducible with different small molecules in mouse cells, looking at stem cell OCT4 expression and development and heterochromatin memory. Another group did something that I thought was just completely crazy, which was they expressed 2 different DNA binding proteins. So they expressed the LEX-A protein or the TET repressor fused to something called a nanobody. A nanobody is a single chain antibody which come from llamas or sharks. They don't require processing through the ER, so you can express them in the cytosol of human cells. So this nanobody binds to GFP, and then they express another nanobody fused to VP16, which also recognizes GFP at a different interface. And so with these 2 artificial proteins, you can have like a GFP sandwich and recruit a domain to a specific sequence of DNA that you can integrate into a cell and control transcription. Again, now 5 different proteins to control transcription. And what this group showed was that if you transfect these constructs into the eye of a mouse and you express GFP from a cell type specific promoter, you can control transcription in different cells differentially. And it's sort of a proof of principle, but it gives you an illustration of how complicated these systems have become and how where artificial synthetic transcription factors have moved towards. I think the only thing that I really want to leave you with from my obsession with artificial transcription factors is that they're modular proteins. They have a DNA binding domain, and they have a domain that confers an activity. These functions are separable, so we can create chimeras that have different binding specificities and different activities, and that a lot of different groups have done this with a variety of applications. And it's been a useful approach to study how transcription works normally, but also to create synthetic systems which can be used to, for things like biosynthesis in yeast, which I didn't talk at all about, or to study eye development in a mouse or what have you. The really big problem with all of these systems is that all these chimeric proteins have a fixed DNA binding specificity. So you have to make a chimeric sequence of DNA which you'll recruit your protein to by either knocking something in or expressing a chimeric promoter from a plasmid. And so this is the real limitation of using Gal4 and TET-R and LEX-A and all these DNA binding proteins that have been used for a long time now. And so really the next big leap forward in artificial transcription factor research was the zinc fingers, which Dana introduced yesterday. So these are programmable DNA binding proteins. They are artificial, but they're inspired by the zinc fingers that are present in natural transcription factors. It's just you can evolve them to recognize an arbitrary sequence of DNA. And so sort of at the same time as people were using ZNF FOC1 fusions for genome engineering, other groups were using ZNF other fusions to control transcription. And so some of the work Dana showed you was earlier than this and some was later than this, but in the late 90s, people started to fuse artificial zinc finger proteins which recognized whatever sequence they wanted to domains to control transcription. And so these are three figures from a paper from Berkeley et al., where they created a zinc finger which bound to the ERB promoter using an ELISA to analyze binding activity and specificity. They then took this zinc finger that they had artificially generated and fused a bunch of N-terminal and C-terminal domains to it, and then targeted that to a ERB luciferase reporter to see whether they could control transcription. And so that's shown on these bar graphs here, and all you really need to see is that this is their control, where you have 40,000 units of luciferase, and if you fuse your zinc finger to a crab domain or this other SID domain, you can pretty much silence expression. Or you can fuse the exact same zinc finger to, again, our favorite VP16 or 4 copies of VP16, which is called VP64, to activate transcription. And so this is really one of the first examples of an engineered artificial DNA binding protein used to control transcription of an arbitrary sequence or of an arbitrary gene. Fairly quickly, though, the zinc fingers, or not fairly quickly, eventually the zinc fingers were replaced by tails because, as Dana said, the tails had much more predictable DNA binding activity. And the tail fusions were used to, again, with the same template from zinc fingers or fixed DNA binding specificity transcription factors, turn transcription on or turn transcription off. I never did any tail work, but it seems, at least from these diagrams, that the cloning was not necessarily straightforward. And these are repetitive proteins which are not easy in all labs to work on. So there were a number of reports that you could clone an artificial tail that would control transcription, but the method wasn't around long enough for it to be widely adopted as a research tool by the average lab. In contrast, I think, to tail FOC1 fusions because there were no other, this was the best way to, the tails were the best way to genome engineer, but there were other ways to control transcription. You can use RNAi to knock genes down, you can use cDNA overexpression to overexpress things, and there just wasn't really enough motivation to generate tail transcription factors. But nonetheless, people did construct these, and it's sort of, the literature is dominated by a fairly small number of labs. But one important thing that people learned from this was that if you want to activate expression of an endogenous gene with a tail or with a zinc finger protein, you frequently need to target multiple tail fusions to the promoter. So everything I showed you with fixed sequence DNA binding proteins, they had artificial promoters that had usually multiple binding sites, so 6, 10, 12 binding sites. If you just have a single binding site, most of the time recruitment of one zinc finger or Gal4 or tail VP16 or 64 fusion is insufficient to activate expression of an endogenous gene. And so people started to clone, or a few labs started to clone larger and larger libraries of tails that were fused to either repression domains or activation domains. And with this, you can see, like, for this gene here, KLK3, each of the individual tail fusions doesn't really activate gene expression that much, but if you combine all of them, now you get pretty good gene activation. And so this is a lesson that we'll come back to with CRISPR in a few more slides. As with the fixed sequence DNA binding proteins, quickly more and more complicated methods for controlling transcription with tails emerged. So one of the methods that I think has been particularly exciting for a variety of fields is the development of optogenetic tools. So these are usually plant proteins that are induced to dimerize with light. So if you express two fusion proteins, one a tail with this CRY2 protein and another a CYB1 protein fused to VP64 in a cell, when you shine a light at a specific wavelength, the two optogenetic proteins will dimerize, and you can recruit an activity to a specific region. So you have your DNA, you have your tail or your DNA binding protein constitutively bound, and when you shine light on a cell, it inducibly brings in a domain that confers some function. And so the data for that is shown here, where they're looking at a reporter and they've got different tail fusion proteins expressed. Actually, this is an endogenous gene. And then you can see that with the right combination of tail optogenetic fusions, you can activate gene expression of a NEROG2, of the NEROG2 gene here. And that's also shown down here where there's no gene expression at T0. When you shine light on these cells, fairly quickly you induce recruitment of the VP64 domain and gene expression. And gene expression requires sustained delivery of light and sustained recruitment of this domain. Because when you withdraw light, you can see that gene expression quickly is reduced. This paper was published actually after all the CRISPRs started to come out. This is from the Zhang lab at the Broad, which a lot of you are familiar with for their CRISPR work. But he was actually one of the early pioneers in tail fusions as well, and sort of where he initially made his name and was hired for, but I think has clearly transitioned to CRISPRs because they're easier to use and easier to design and create. But the same group in this paper, they've had a long-standing interest in studying neurobiology and autism. And so they used these optogenetic inducible tails together with an AAV viral system to deliver the tail fusion proteins to the brain of a mouse. And so you can stereotactically inject AAVs into the brain where they'll invade, express your two fusion proteins, and when you shine light onto the brain of the mouse, you can see you get some increase in gene activity, although it's relatively modest. But nonetheless, I think this provides a first proof of principle that these kind of tools can activate expression of endogenous genes within living animals and are not just sort of toy proteins to be used in, cancer cells in a dish, but we might actually be able to learn something about, things as complicated as brain development or learning or memory, which I think would be very, very cool. In the same paper, as was done for the fixed sequence DNA binding artificial transcription factors, this group also fused a variety of histone modifying activities to a tail or to the SIB portion of their tail optogenetic system, and showed that you could either increase or decrease expression of a gene by recruiting a specific histone modifying activity to a specific locus. And so I think this is a common theme throughout all of this, is that people would like to be able to deliver any domain to a specific sequence of DNA, and how you do that has evolved over time, but the basic motivation is to develop systems where you can deliver specific activities to specific regions of DNA to control transcription. Similarly, you can actually not modify histones, but actually modify DNA itself. So if you make a tail fused to a TET protein, you can induce demethylation of DNA, which activates expression of imprinted genes. So these TET proteins catalyze the oxidation of methylated cytosines to hydroxymethylated cytosines, which eventually leads to demethylation of DNA and activation of gene expression. So they showed that you can recruit this tail to a specific region of the genome where you induce a relatively modest demethylation of this sequence of DNA, but that this demethylation leads to increases in expression of the gene. So we can either directly with tails, now people have shown directly with tails you can recruit either basal transcription factors, you can modify chromatin, or you can directly modify the DNA to influence the output of the genome. So as with the fixed sequence binding artificial transcription factors, the caveat with all this research was that this kind of, the creation of these kind of systems was, a lot of work, because cloning, it was a lot of cloning. Not all the fusion proteins work. You need to clone multiple fusion proteins to activate gene expression. The proteins are repetitive. You can't package them into lentivirus. And the whole thing was an interesting exercise in, can this work? But really is not, unless if you have a very specific interest in a specific gene, is not something that people were jumping into, without a really quite significant motivation. And so that brings us to CRISPRs. So I think with both genome engineering and controlling transcription, CRISPR has exploded relative to any other method, and this is largely because it's quite easy to use. And so, as has been stated several times here, if all you have to do is express a different small RNA to target a nuclease or an activity to a specific region of the genome, that's very, very easy relative to re-engineering new proteins every time you want to target a new gene. So I won't reintroduce the nuclease. We've belabored that. Inspired by some of Martin, Janek, and Jennifer's work from the very first science paper, we at UCSF have been interested in whether we can use a nuclease deactivated version of Cas9, which we've called dCas9 or dead Cas9, as more than a nuclease. So as if the nuclease wasn't already cool enough, can we do more stuff with it? And so there's been a really neat collaboration between, 5 or 6 labs at UCSF over the last 3 years where we've shown that we can use dCas9 in E. coli, yeast, and human cells to do a variety of things. And so this really was started by Stanley Shi, who's now a professor at Stanford. He was a grad student at Berkeley where he attended the Doudna lab meeting. He was in the Arkin lab. And he was a synthetic biologist or a physicist really by training. And he started as a fellow at UCSF at the same time I started as a postdoc, and he had this crazy idea of using dCas9 to control transcription, or just generally to see what he could do with it. And so what he showed very quickly and elegantly was that in E. coli, and I'll go through the data in more detail, you could use dCas9 and bind it to the genome, and when positioned appropriately, dCas9 would block an RNA polymerase elongation, thereby silencing a gene. I got involved when we started working in human cells because he didn't have the technical expertise, and so together we showed quite quickly that you could use dCas9 fused to a variety of domains to either activate or repress transcription by delivering regulators to specific regions of the genome. And then in collaboration with Bo Huang's lab, Stanley and I showed that you could, together with Bao Hui, that you could use a GFP dCas9 fusion to image the genome in a living human cell. And so I'll go through all this in more detail, but that's generally what I'm going to talk about. And then I'll talk about additional things that we've worked with the Wendell Lim's lab and Ron Vale's lab to sort of ramp up our ability to control transcription and image the genome. But it's been fun to work in this collaborative team at UCSF. So the very first experiment Stanley did actually failed. So he asked whether you could express wild-type Cas9 with a guide targeting the E. coli genome in E. coli. And predictably, this killed the E. coli. So the next experiment he did was ask whether you could use catalytically inactive Cas9 to bind the E. coli genome, and if so, in a living cell, what this would do to transcription. And so he had the insight, and I think we've continued in this vein to start always with fluorescent reporters, which are incredibly useful tools for troubleshooting anything new in synthetic biology. So he targeted dCas9 downstream of RFP reporter that's expressed from an integrated locus in the E. coli genome, and expressed either three guides targeting the template strand or 3 guides targeting the non-template strand. And surprisingly, he saw that you could repress transcription of RFP, but that this only worked if you bound it to the non-template strand, where targeting the template strand seemed to have no effect on impeding RNA polymerase. And what was really remarkable was that was not, I think, so much that this worked, period. I mean, I think it was expected that Cas9 would still bind to its target site. And people with other methods have shown that if you bind a protein just downstream of a transcription start site, you can impede RNA polymerase. But really, it was some property of Cas9 which really robustly blocks RNA polymerase elongation. And so it was really the effect size, I think, that was astonishing to us, where you can get well over 100-fold repression of RFP transcription if you bind Cas9 to the right spot downstream of a transcription start site. And so, as with everything, activity doesn't really mean anything unless if you have specificity as a tool, at least. And so Stanley, together with Matt Larson, who was a postdoc in Jonathan's lab, did an RNA-seq experiment where they again targeted RFP and asked what happens to the rest of the transcripts in the cell. And what you can see just generally is that RFP is repressed. This is a log 10 scale, so it sort of doesn't quite do justice to how much repression you have here. But nothing else in the cell changes that significantly. Matt went on to show how this worked mechanistically. So using a technique developed in the Weissman lab called NetSeq, which can measure where along a gene the elongating RNA polymerase is bound or resides, with a pull down, Matt showed that in the absence of a guide RNA, you could see an elongating RNA polymerase along the gene body of this reporter. But if you targeted Cas9 right here, and the dotted line is shown at the 5' nucleotide of the guide RNA, RNA polymerase would initiate transcription, hit the edge of Cas9, and just completely stop elongating. And the distance between the edge of the 5' guide and where this peak of piled up RNA polymerase is 19 base pairs, which is thought to be the footprint, the exact footprint of RNA polymerase between the site of base addition and the edge of the protein. Strongly suggesting that Cas9 was blocking RNA polymerase elongation and disrupting transcription. Stanley showed...
Could I ask you, I'm still stuck on the strand specificity.
Yeah, so am I.
So where's the PAM? On the slide.
Yeah.
So on one strand, the PAM is downstream, and on the other strand, the PAM is upstream of where the guide is
Yeah.
binding. So I'm just wondering, so if it's just Cas9 being there that's inhibiting elongation, you'd think either one would work fine. But if it has to do with what part of Cas9 the polymerase runs into, or whether it's whether you've got the displaced strand from the hybrid to move through along, that would make it.
Yeah, it would be quite significant. Those are as good of ideas as I've heard anybody propose. I don't think so it's not that targeting the template strand has no effect. You can get about a 10-fold reduction, and because this is plotted on a log 10 scale, it looks close, but there is some reduction in transcription. And the reduction in transcription from targeting the template strand is about as much as reduction in transcription as you see in human cells. So in human cells, the RNA polymerase is either more processive, which is likely the case, and so maybe there's I don't think Stanley. We have talked about this with a lot of people, and nobody understands it. And so I don't know. It is what it is. At the end of the day, it's not quite as black and white as presented here. There is some decrease in transcription if you target the template strand. And I'd love to. The ideas you raised are as good as any, and we haven't tested them though. So.
The paranicases sort of have the same mysterious behavior, how they only work in one orientation, where the hands are out. And we never knew what that was due to. We thought that maybe it has something to do with DNA distortion, like directional DNA distortion. Like maybe it's distorting DNA out, and they can both still bind. So if there's some sort of directional distortion of the DNA, maybe the polymerase hits that, pops off. Whereas if it's the other way, it can come along and knock it off. I don't know.
Yeah, I think we'd have to. I think to me, without having given it much thought, the easiest way to understand this would be to crystallize RNA polymerase with DNA and like all that. But that's nearly impossible probably. And I don't know. Probably Jennifer's lab might have another idea, but because there's no preference for strandedness in human cells, and I'm more interested in human cells than E. coli, I've been reluctant to follow up on this. Stanley also showed, though, and this is, I think, an important point and has interesting implications, that you could use Cas9 to compete with binding sites for sequence-specific transcription factors or general transcription factors. So if you bind Cas9 to different spots along the promoter of this gene in E. coli, like especially this P1 binding site where you're blocking the -35 element, you could quite strongly decrease transcription. And so this implies that it's possible that in any organism, if you express the right amount of Cas9, you could actually map along a promoter or an enhancer the regulatory regions that are required for expression of a gene. And this is not something that has worked particularly well in human cells, but I don't think we've tried particularly hard either. And I think if this worked efficiently in human cells, could be a really interesting way to map, to functionally map where the binding sites are that matter for transcription of a gene by competing with, for binding with dCas9. Stanley went on to show that this control of transcription was inducible and reversible. So this is TET-inducible promoter that drives expression of DCAS9. If you, it's a TET-on. So when you add doxycycline, Cas9 is expressed. Following expression, you see repression of this reporter activity. If you remove doxycycline and Cas9 is degraded, repression comes right back. And so this was, good proof that we weren't doing anything permanent to the genome. The DCAS9 is actually nuclease inactive. It's not making mutations that are, preventing protein production. But also suggests that this could be an interesting tool for dynamically controlling transcription of any gene. He showed in E. coli that you could get synergistic repression of gene expression and that you could target 2 genes. So in this experiment, he's targeting 2 reporters, one an RFP reporter and one a GFP reporter. And you can see when you express a guide targeting GFP, you lose GFP expression. If you target RFP, you lose RFP expression. If you target them both, they're both gone. So for better or worse, science publishing is what it is. And when he submitted this beautiful piece of work, the reviewers asked, so what? They said, essentially, E. coli doesn't really need new genetic tools, shows that this works in either a eukaryote or a biofuels bacteria or a pathogen or something that needs better genetic tools. And so he very quickly started collaborating with a lot of people, including people at Berkeley and us at UCSF, and we asked whether we could show that this would work in human cells. And I'll show you a combined experiment that was actually done later. Well, I'll show you this experiment first. So the first experiment we did was we targeted a GFP reporter that was integrated randomly into the human genome, where we have GFP expressed from an SV40 promoter. And then we just expressed a bunch of guides with DCAS9 alone and asked whether we could silence GFP expression. And you can see in the light bars that you do get some silencing. This was a huge disappointment relative to E. coli, where you get over 100-fold silencing. But we could show that you get, for a number of guides, roughly 2-fold silencing. This was quite unsatisfying, though, so we wondered very quickly whether we could fuse additional domains onto Cas9 to more potently control transcription. And so I'll just show the one that has worked the best, but we fused a C-terminal crab domain to Cas9, and in the same experiment, target a chromosomally expressed GFP reporter with a guide targeting GFP, and we can see that we get about 20-fold repression of GFP expression in this experiment. And actually, the bulk of the cells look GFP negative, and there's a small tail that contributes most of the signal left in the sample. That's further summarized here for a number of guides which compare repression if we use DCAS9 to repression if we use a DCAS9 crab fusion protein. And so you can see that some of the guides don't work, some of the guides work quite well, but that in most cases, there's some silencing with DCAS9 where we're inhibiting RNA polymerase elongation, but that the addition of this crab domain, which is a transcriptional silencer, improves our ability to silence expression of this reporter gene. And we showed early on that this also worked at endogenous genes. So we silenced 2 cell surface proteins and used fluorescently labeled antibodies and FACS to measure expression. We showed we could silence the transferrin receptor or a CXCR4 HIV co-receptor. And this worked, but clearly is not perfect. We're getting 80-ish% repression. We showed we could silence both genes at the same time, implying that we had some activity at endogenous genes and that this could be multiplexed. To show that we actually were getting silencing dependent on the crab domain rather than just on some kind of an altered fusion protein that was competing with the polymerase, we targeted sequences upstream of the promoter. There are redundant binding elements within this promoter, so we didn't necessarily expect that we would get competition with fixed sequence transcription factors which would silence the gene, but rather here our expectation is that the crab domain, when positioned upstream from a transcription start site, recruits additional proteins to silence expression, and we saw that targeting upstream the promoter, we could get a variety of guides which would decrease GFP fluorescence. again, supporting the idea that we have two types of activity in this kind of silencing. We showed that this worked broadly in yeast. And then in yeast, I think what we were able to show, but we've been sort of unable to show efficiently in human cells, we showed that we could use dCas9 to compete with a transcription factor for binding sites inducing silencing. So here we have an artificial promoter with 7 TET-O sites upstream of a venous reporter. And if we express the RTTA, we see that the reporter gene goes on. And if we co-express the RTTA and dCas9 with a guide RNA that targets the same TET-O binding sequence, we see that dCas9 interferes with the ability of TET to bind to its operators, suggesting that we can use dCas9 to map cis elements and promoters, in yeast at least. Repeating Stanley's general workflow, we wanted to know how specific this would be in human cells, and so I did an RNA-seq experiment with the same GFP reporter cells that I showed you just before, where we can express our Cas9 crab construct and a negative control guide RNA, or our crab construct and our guide RNA targeting GFP. And you can see from this plot that GFP is the only gene whose expression changes in this experiment. And so I realize this data looks almost fake, but we did it twice, and it is what it is. I guess I'm good at RNA-seq or something like that. But no, this was really what made us very, very excited about this method. So Jonathan's lab had spent 5 years developing really quite sophisticated RNAi libraries, but the problem with RNAi has always been the off-target effects, especially in screening. And so this suggested to us that we could develop an orthogonal system that was like RNAi, but that had quite reasonably good activity, even with a very limited number of guides that we had tried, but that would be exquisitely specific. And this, I think Jonathan will touch on in his lecture tomorrow, is especially crucial for the creation of genetic interaction maps, where your off-target activity becomes more apparent the more pairwise comparisons you make. And then we did some more RNA-seq to show that expression of just our dCas9 constructs or a guide RNA alone doesn't have any effect on our GFP reporter, and that also there's no gross signature of toxicity from doing these kind of experiments in human cells. We've gone on to clone genome scale libraries and use this as a functional genomics platform, but Jonathan and Max will talk about that tomorrow, so I'll just touch on 2 additional things that we've shown with CRISPR-I that we can do in a low-throughput assay. So as with the TET-on, TET-off vectors, it's useful to be able to dynamically control transcription. And so we cloned a TET-inducible CRISPR-I construct, which we've shown really lacks leakiness, but is nicely inducible. So this is a 7-day experiment, 9-day experiment where we've targeted a bunch of essential genes, or we have some negative controls. And you can see that in the absence of doxycycline, these genes don't deplete from the population. But when we add doxycycline to turn on our CRISPR-I fusion, we see that essential genes are depleted and the cells are removed from the population. And you can get down close to only 1% of the cells remaining over a 9-day period, which suggests that the system is pretty efficient. We also showed that repression of gene expression is reversible. So again, we're not making a permanent change to the DNA, and we're not inducing a permanent chromatin state either. But rather, if you knock a gene down and then remove doxycycline, expression comes back, shown here. I think maybe most excitingly to me, we've shown just with a small handful of guides that we can use this system to repress non-coding RNAs. And so I think Cas9 is an amazing tool, but one of the things it's not particularly well suited to do is study RNAs that lack an open reading frame. So the way most people are using Cas9, you generate an indel to knock out a gene, but if you don't have an open reading frame, that's not going to do much to a non-coding RNA because the indels are fairly short. So you could use Cas9 to paired nicases or dual nucleases to delete non-coding RNAs. But we know from tail and zinc finger work that this has resulted in a lot of artifacts in the non-coding RNA field, because frequently you also delete promoter elements or enhancers or other regulatory features that are contained within these non-coding RNAs. And so a way to specifically control expression of a non-coding RNA but not neighboring transcripts or perturb enhancer functions, I think promises to reveal in an unbiased way what some of these things are doing. And so what we've done is shown that we can target these 6 non-coding RNAs, which are pretty well characterized, and knock them down. I think we said something like most of them we knocked down by at least 85%. This is a log scale plot, so 90% repression is right here. This one we don't do a particularly good job on, and there's a couple inactive guides, but for the most part we can repress things by 90 to 99%. And this holds up if you look by fish, which is shown over here, where if we do a fish experiment looking for expression of exist RNA, which coats the inactivated X chromosome in female cells, we see that there's no detectable exist by fish, really suggesting, I think, that the majority of our qPCR signal left here is probably off-target activity because we're not using TACMAN probes. One thing that we noticed early on, and that I was actually called out by Jennifer in her first lecture, is that in the original iteration of the sgRNA design, and the design that likely most of you are using for nuclease experiments, you're using a truncated stem relative to the wild type. And so, at least for imaging and for controlling transcription, we noticed that guide RNA expression limited function. And so we went back and made a series of changes resulting in this construct down here, where we restored the wild type stem that's found in pyogenes and took out a Pol III terminator right at the beginning of the stem sequence. And this results in increased guide expression and somehow more stable guide probably folding, which improves loading into Cas9 in cells. And the experiment that Jennifer showed, I would, to defend myself, would not have predicted that my design would be better in her experiment, because she's pre-assembling in vitro transcribed RNA into Cas9, and so it doesn't have a chance to be influenced by the RNA remodeling proteins that are present in human cells. But we showed that in human cells, this does actually improve things. So if we take a guide RNA in our GFP reporter assay that has some modest activity, with our improved guide, we get about a 5-fold increase in activity. And even more importantly, we see that with single copy lentiviral integration, there's no relationship between guide expression and activity, which was really important for us thinking about screening, where we rely on having single copy integration for our experiments. So we also, copying the zinc fingers and tails and fixed DNA binding proteins, wondered whether we could activate gene expression. And so we copied what everybody had been doing and actually used the same Gal4 UAS elements upstream of a GFP reporter and cloned a guide RNA that targeted these Gal4 UASs, and then expressed a Cas9 fused to VP64 and asked whether we could turn on GFP expression. And in this experiment shown here, you can see that we get about 20-fold induction of this GFP reporter, which is reasonably good. Disappointingly, though, we knew when we target with a single guide RNA most endogenous genes, we really fail to activate expression. And so this is not a particularly surprising result given the literature from the tail field, where you require 5 or 6 binding events to activate expression of an endogenous gene. But nonetheless, it wasn't a fun result to see. And so I had a very fortuitous conversation where I was helping Marvin Tannenbaum, who's in Ron Vale's lab, design some guides to delete a gene that he was interested in, and I asked him what he worked on, and he, other than the project he was telling me about, and he said, I designed this crazy system where I can express an epitope on a protein and then target a single chain variable antibody to this epitope and induce protein-protein interactions in cells, and you can express multiple epitopes and recruit many of one copy of a protein to a single protein. I said, that sounds perfect. And so we expressed together a dCas9 that's fused to 10 copies of this highly optimized epitope, separated by some linker regions. And then in the same experiment, if we express the same guide RNA targeting CXCR4 that I showed you on the previous slide, where before with our dCas9 VP64 fusion, we don't get very much activation. Now with 10 copies of VP64, we get robust activation, and it's even across the population. So the FACS plots are shown here, where this gene is really off in these cells. So this is an unstained control. This is a stained control expressing a negative control guide RNA. And this is a guide RNA population where we're activating CXCR4. And you can see, well, two things. The negative control stained sample looks like the unstained sample, so there's really no detectable protein expression here. And then the population, when you activate CXCR4, homogeneously shifts by about 40-fold up. This is Cas9 is a competitive field, and so there are to be fair, other approaches out there that have thoughtfully sought to activate expression beyond just VP64 fusions. And so two recent ones I'd like to highlight are from George Church's lab. They've developed a tripartite activation domain where they have VP64, P65, which is a NF-kappa B transcription factor activation domain, RTA fusion. RTA is a Epstein-Barr virus protein domain. Their published work seems to suggest they still require more than one guide to efficiently activate transcription of endogenous genes. The Zhang lab at the Broad has also published a chimeric activation system that they've called the synergistic activation mediator. Their system fuses VP64 to Cas9 and then uses the guide RNA to recruit two MS2 proteins. So the MS2, I'll talk more about the MS2 MCP protein in a couple slides. But suffice to say, with this system, they can recruit 4 copies of, or 5 activation domains total. So they recruit 1 VP64, 2 P65s, and 2 HSF1s to a locus with 1 guide RNA. And this seems to work quite well. They get significant increases in transcription for a variety of genes. One thing I'd like to point out, though, in doing these experiments is fold change is not really a very good way to measure this. So if your gene's not expressed, you're dividing by 0, which gives you a very large fold change. And to their credit, this paper and other people have started to display more like an RPKM number where you have a quantitative measurement of fold increase. But even there, it's difficult to know how to normalize these things for genes that are really not expressed. I've compared their published data to our own unpublished experiments for the Yamanaka's stem cell factors, the OCT4, KLF4, SOX2, and NANOG. And it's actually sort of amazing. The fold changes are very similar for the two systems, implying that there's some intrinsic limit to activation that both systems are hitting. And it's possible that you require for true, activation of a gene, recruitment of different types of enzymatic activities other than just these acidic activation domains. So you may require a acetyltransferase or a demethylase to truly activate a gene like would occur during development. Okay, so what is the MS2 MCP system? Because I'm going to move into another paper that uses this extensively. The MS2 MCP system is a system that has been used since the late 80s. MS2 is a stem loop sequence from a phage genome. MCP is the MS2 coat protein. So it binds the phage stem loop and coats the phage genome. And it's been used by predominantly Rob Singer's lab, but a variety of other labs to track mRNA localization within living cells. And what the Singer lab and others have shown is that you can express many MS2 fusions on an mRNA and then express an MCP GFP fusion and visualize the localization of an RNA within a living human cell, or a yeast cell for that matter. So here's a yeast cell. Here are 3 human cells, and you can see that you get co-localization with whatever markers they're looking for here. with the GFP signal suggesting that this can be used to visualize tagged RNAs in a cell. This is not the only RNA stem loop that has been used in synthetic biology. So this is from a review that was talking about some of the best characterized stems. Their RNA sequence is shown here, and then the critical protein binding interfaces is shown in blue. And I think what's notable is that there are more than one of them and that they're orthogonal. So the MCP protein binds MS2 with an affinity of about 0.4 nanomolar. The PCP protein binds the PP7 stem loop with an affinity of about 1.7 nanomolar, so not quite as tight, but binds to the MS2 stem loop with a KD of about 1 micromolar. So they're very specific for their target stems, which implies that they can be used orthogonally. So I was involved with a project that came out this year with the Lim Lab, where we asked whether we could use the guide RNA to encode both the local, to guide Cas9 both to a location and to recruit a defined activity. So use the guide RNA as a two-part thing. So everything that we've done with protein fusions, you have a guide RNA that encodes targeting information, but then the effect of Cas9 is encoded on the protein. So with one Cas9 or one dCas9, you can only do one thing. You can only either inhibit or activate expression, but not both. Where if you could encode the activity on the guide, if you can encode both the localization information and the effector domain information on the guide RNA rather than on Cas9, with the same Cas9, you could both activate and repress transcription. And so that's laid out here, where using two different stem loops, you can target Cas9 to two different genes and recruit two different RNA binding modules that bring along two different activities. And so the goal here is to create a complex transcription program with one Cas9. So you could do this with two Cas9s fused to two different things, or you can do this with one Cas9 with two different RNA stem binding proteins. Oops. Yeah. So this is really mostly work by a really talented postdoc, Jesse, who's now at the University of Washington. But he showed that, and this experiment is in yeast, that you could express dCas9 and an sgRNA targeted to this reporter, where you have one binding site upstream of a venous venous protein or mRNA, sorry, venous encoding sequence, and then co-express either an MCP, PCP, or this other RNA binding protein COM fused to VP64. And when you co-expressed all three of these things, you could activate expression of your venous reporter. And the level of activation seems to vary between these things. And this is not quite fair. This is the dCas9 VP64 fusion, the direct protein fusion. Somehow in yeast, this really works quite poorly. It's not clear to me why, but at least in yeast, this is dramatically better. It's dramatically better to recruit the activation domain with the RNA than it is to directly fuse it to Cas9. So now that he has three working stems and stem binding proteins, he asked whether they were orthogonal. And so what he did was expressed either the guide targeting, in this case, a reporter with 7 binding sites, and co-expressed in each combination shown with these lines, the stem and its either cognate binding protein or the orthogonal binding proteins. And you can see from this venous expression, this is all FACS data, that when you express a guide RNA with a stem loop and its cognate stem binding protein, you get activation of transcription. But when you express these other stem binding proteins, you don't get significant activation of this reporter, suggesting that these can be used orthogonally together in the same cell. He also showed that 2 copies of the stem are better than 1, so you can recruit 2 proteins with 1 guide RNA. So that's shown here for MS2 or PP7, or yeah, PCP. He also showed that you could create a chimeric stem where you used where you had a guide RNA that was that had both an MS2 and a PP7 stem on it, and that this could additively or synergistically induce transcription by recruiting 2 different proteins, both fused to VP64. This also works in human cells, although not quite as well, where he showed that you could both activate and repress a protein within a human cell, or I showed this, you could both activate and repress a human, 2 genes within a human cell. So here you're recruiting VP64 MCP to CXCR4 to activate expression of CXCR4, and with the same dCas9, you're recruiting CRAB fusion protein to this gene B4-GalNT1. When you express a guide targeting CXCR4, you see an increase in expression. When you express a guide targeting B4-GalNT1, you see a decrease in expression. And when you express both guide RNAs, you see that CXCR4 is activated and B4-GalNT1 is repressed. Back to yeast, the Lim Lab has a longstanding interest in metabolic engineering in yeast, and so they wondered whether they could manipulate an artificial metabolic pathway and both activate and repress transcription of different genes to execute a complex synthetic program which could control production of different biosynthetic molecules. And so what this is a 5-gene pathway that produces a compound called violacin that's purple. So Vio-A is the initial gene in the biosynthetic pathway, then B, then E, then there's a branch where if you express Vio-C, you produce this molecule. If you don't express Vio-C or Vio-D, you produce this molecule. If you do express Vio-D, then you express this. If you express both Vio-D and Vio-C, you express actually violacin. And so the goal was Jesse created a yeast strain where B and E and D are on, but A and C are off. And so in this case, you don't express anything because A is off and this biosynthetic pathway isn't initiated. And the goal was to see whether he could activate or repress A, D, and C and manipulate the pathway in different directions to produce either this, or this. And so I'll just cut straight to the experiment. In the absence of any guide RNA expression, the pathway is off because violacin A is not expressed. The yeast are white, and there's a little bit of leaky expression of the PV product by HPLC. If you activate expression of A, the biosynthetic pathway is turned on, and you produce PV and a little bit of PDV. If you activate expression of both A and V, you start to produce violacin, where if you inhibit expression of, what is this, V, now you shift the cells toward PDV away from V. And if you additionally activate expression of C, now you produce both PDV and DV, suggesting that with a synthetic program, you can control this pathway pretty much however you want and produce one or the other compound depending on what you would want to produce. So I think this is a bit of a toy example because nobody cares that much about violacin, but at least is proof of principle that you can control expression of 3 genes in yeast and predictably do some metabolic engineering. So I think some questions that will partially be answered tomorrow by Jonathan and Max that relate to synthetic transcription control with CRISPR are, can we use CRISPR-I and CRISPR-A to manipulate pathways in yeast? Like if you could imagine manipulating a signaling pathway like the EGFR pathway where you would, or a RAS pathway where you could turn on and off different GEFs and GAPs, or different MAP kinases, or expression of different transcription factors, and manipulate cell activity in that manner. Can you use CRISPR-RNA for functional genomics to define the function of the many transcripts encoded by the human genome, both protein coding and non-coding? And then can we use CRISPR-RNA? Well, I already said that one. And then I think maybe most interestingly is, I think it would be very cool to use CRISPR-A especially to search for combinations of genes that control complex phenotypes. And so the best recent example of this, but there are older examples, is 4 gene combinations of transcription factors that govern reprogramming from fibroblasts to stem cells. And I think this is just the first example of, there was essentially a cDNA screen to find a combination that controlled dedifferentiation into an iPS cell, but we can potentially use CRISPR-A for these kind of screens much more easily. Work not from UCSF, but from other people has shown that dCas9 can be used to manipulate chromatin. So from this year, from the Gershbach lab, there's been published a dCas9 histone acetyltransferase fusion. So the minimal hat domain was fused to dCas9, and they were able to show that when you target this to a gene, it acetylates the local chromatin, inducing gene expression. And I thought especially cool was they showed that if you target multiple guides to an enhancer region that's far from a gene, in this case the hemoglobin S2 enhancer, you get activation of genes that are very far away from the gene, suggesting that acetylation of an enhancer induces looping, which activates expression of a gene. And that's shown by ChIP-seq here, where you pull down on H3K27, and you see you're targeting guides up here to this HS2 enhancer, and you induce H3K27 acetylation, but that even though your Cas9 fusion is targeted here, if you look at the, what is this? HBE promoter or HBG promoter, you see that you also are inducing H3K27 accumulation at the histones at the gene promoter, which induces activation. Similarly, another paper has shown that you can fuse dCas9 to a histone demethylase and use this to turn gene expression off. And so we use the CRAB domain, which is a domain that acts to recruit the CAP1 scaffold and a whole bunch of other things. In our hands, it works as a short-range repressor, so that if we target it distal from the transcription start site, it has no effect. This group showed that that was more or less true as well, but also they showed that their dCas9 LSD1 fusion, when targeted to an enhancer domain shown here with this ODE1 label that's quite far from the transcription start site of OCT4, you could turn OCT4 off, implying that demethylation, in this case, demethylation turns transcription off. So they're removing, methylation is complicated, right? It can either increase or decrease transcription. And in this case, they knew that demethylation should turn the gene off. And they showed that here, where if you look at not the methylation mark that they're removing, but a different methylation mark, you don't see loss of H3K4 dimethylation if you're using the LSD1 fusion at the enhancer, but you do see H3K27, I can't read that. Let's try. Anyways, basically they showed that the chromatin marks that change make sense. So I think transcription is very complicated. There's a lot we don't know. In my opinion, using artificial constructs to control transcription teaches you a lot about how it works normally. I think currently, mostly we're doing this, where we have very simple systems to control transcription. Transcription is, as I hope I've gotten across, a very complicated thing. And if we have the appetite for it, I think we now have the tools to construct artificially things that look more like this, where we're using activators and acetyltransferases and methylases in combination to control expression of endogenous genes. And so I'd like to end with just 5 minutes of some additional applications of our CRISPR RNA-guided DNA binding platform. And so what we've shown at UCSF is that you can use dCas9 as an imaging platform to image arbitrary sequences of DNA within a living human cell. And so this is, at its most basic, shown here, where we have directly fused GFP to dCas9. We can then express this at very, very low levels from an inducible promoter where we're not activating the RTTA, and we co-express a guide RNA. And if you target a repetitive sequence like the telomere, so here's the telomere repeat, TTAGGGT, and here's a PAM, and here's a protospacer, you can see that our improved guide RNA nicely labels the telomeres, and we showed that this correlates quite nicely with fish. This is, again, our original guide RNA, which works quite poorly, and the improved guide, which is better expressed and more robustly loaded into Cas9, works very efficiently to label telomeres. I think the coolest thing is seeing the actual movies. So this is a movie of a human cell with dCas9 bound to the telomeres, and you can see they don't do a whole lot. They sort of wiggle around, but yeah, I think this is the kind of thing you can do. I think what we're limited currently by is if you have a GFP dCas9 direct fusion, you have to have at least 40 to 50 binding events to see a spot. And that we got around by showing in proof of principle experiments that if you target these MUC genes, these are mucin genes that have a natural repeat within the human genome, that there's either, like, depending on the exact region you target, there's 100 or 200 repeats. You can see discrete spots. We initially saw 3 spots, and we were very confused by this. We karyotyped our cells. They're triploid for this chromosome, so that made sense. And that this signal correlates really nicely with oligo fish. And we also show that you could target 2 spots adjacent to each other on a chromosome. So here you see 2 fluorescent spots next to each other, and there's 3 copies of that chromosome.
Are there plans to hook up the sun tag to the dCas9s?
We are very predictable at UCSF. We repurpose old stuff and just use it with CRISPR. So yeah, can you recruit multiple fluorescent proteins to one dCas9? So Marvin and I immediately did this. We fused, Marvin fused this GCN4 epitope to a variety of proteins, but I'll show you that we've fused it to dCas9. The next answer to your question is, can we do single molecule imaging, is no. The background is too high still, and so you can do single molecule imaging with this system where you can recruit 24 GFPs, but it requires that you have your unbound protein being diffusing quickly so you can motion blur out the background is really the answer. So this single chain variable fragment binds to this epitope really tightly. KD is about 44 picomolar. If you do fluorescent bleaching experiments, the half-life of GFP bound, or the antibody bound to the epitope, is about 5 to 10 minutes, which is pretty tight. This is an experiment imaging kinesin moving along microtubules. And you can see that the background unbound kinesins move very quickly and don't look directional, where the kinesins that are walking along are moving directionally across the cell. And so this is not a Cas9 related application, but I think is a pretty cool way to study motor proteins. And if you take the max intensity projection of the brightest 50 frames and merge them, you see that you can actually trace individual kinesins moving along a microtubule. You can also use a kinesin mutant defective for ATP hydrolysis that now just binds to microtubules but doesn't move directionally to study microtubule dynamics with the SUNTAG system. So broadly, microtubules aren't that dynamic in this cell, but if you look closely, there are on the right, you can see individual microtubules extending directionally. And this is really all work from Marvin. But you can also use this fused to Cas9, where we again used our same telomere targeting sgRNA and targeted telomeres. And you can see before where you can see some spots, now you see really bright spots. And we used 24 copies of the epitope, and we see on average about 20-fold increase in fluorescence, suggesting that the majority of the epitopes are occupied by a single chain GFP fusion. And so that's it. Thanks to a lot of people, and thank you for your attention.